Today we have Naama Lang-Yona from the Technion, Civil and Environmental Engineering. And she's doing atmospheric microbiology. And before Technion, she was at Max Planck, Germany, and she did her PhD at Weizmann. And take it from here.
Thank you very much, Nata, for the invitation, and it's always great to be at the Hebrew University, and here specifically. I don't know, you are a geologist, atmospheric scientist, oceanographer. Good. So it's really nice to be here, and I know you are a very diverse audience, so I prepared my slides to be as broad as possible, but if something is not clear, please stop me. I tend to speak a bit fast, so feel free to wave and ask questions. So today I'm going to present a study, a topic that we study in our lab on the dust microbiome. I call it the Dustospheres because we find a very rich community of microorganisms that live on this dust. And apparently not just transported from one place to the other, but really viable and active. And maybe they can do some very interesting things that we still try to find out and figure out the extent of this phenomenon and how its impact global cycles and our Earth. So let me start with a brief introduction on atmospheric aerosol in general. Probably most of you know atmospheric aerosols are solid or liquid particles suspended in the atmosphere, and they can originate primarily from many different natural sources, right? We have the dust and the volcano ash and biogenic pollens and fungal spores, marine aerosols. They can also be emitted from anthropogenic sources like combustion and fires and vehicles and etc. Aerosol could also be formed in the atmosphere, and then we call them secondary atmospheric aerosols, like volatile organic compounds emitted to the atmosphere, getting oxidized, photo-oxidized with UV, and then form nuclei of aerosols to form the aerosol in the atmosphere. So basically, bioaerosols, the topic of this lecture, is mostly these ones, right? Pollens, fungal spores, etc. Apparently, they are more abundant than we thought. They could also be in the marine environment, like fragments of algae or even bacteria coming from the ocean, viruses lifted up to the atmosphere with this marine salt. Also from dust, as we will see today. Also from volcano and very extreme environment, they can emit also microbes with these particles. So microbes can live in very extreme environments, hot spring, volcano, a very salty environment, but also in the atmosphere. And today, this is what I'm going to show you, that the atmosphere is not sterile at all, and it's very active microbiologically. And in this review that was led by Ariel here, we discuss this cycle of bioaerosols, the different sources of these microbes to the atmosphere, how they go, what the interaction with different ingredients in the atmosphere. So clouds would be the first thing I can think about, but also UV, pollution, CO2 increase. All this stuff could interact with those microbes when they travel in the atmosphere for days. And then how that impacts them and how they impact them when they go back to the land. Would they survive? Would they be modified? How would that affect their impact when they are landing back to Earth? So this is something we are really interested in exploring, the processes that occur to microbes while they're in the atmosphere, how that impacts their activity and their contribution to global processes. Another thing we saw, you can see in this figure, is a bit like a yearly interest in bioaerosols. So you can see that in late 80s, started to increase the number of papers published on bioaerosols, mostly on fungi and bacteria in the beginning, less so on viruses, archaea, and others. Here you can see a really increased sharp peak with the corona, of course. Viruses are airborne, and then we have really peak of virus studies increasing and also highlighting, shedding light on this topic of aeromicrobiology. So in general, we find many new instruments for studying bioaerosols, detecting them, sampling them. So it's really good. It's really nice for us. And we think that this is going to increase further. Sorry. In the group, we do a lot of things, and here I highlight the main things, the main topics we study. We have the ocean-atmosphere interaction studies, which is very interesting to see emission of pathogens and the transfer in the atmosphere and then impact on algal blooms. We try to understand the role of the surface microlayer in this emission, etc. We have studies on antibiotic resistance that goes to the atmosphere and transferred through the atmosphere, so sources like wastewater treatment plants and hospitals could be hotspots for antibiotic resistance, and these genes could go to the atmosphere, and we try to understand their distribution through the atmosphere. Another study we do, which is not related to the atmosphere, but with our lab pets, with algae, we try to see a way to biosequester carbon using liquid core beads. And the study we highlight today is the dust-borne microbes, and this is what I want to show you today. So why dust? Probably this is something most of you know. Dust is a global transporting vector, really projected to increase with global changes. Crossing ocean, as you can see in this figure. I hope you can see this dust plume in the Atlantic. And it's a key vector for transferring microbes, basically hitchhiking on these dust particles throughout oceans, throughout continents, and this is a significant land path that they transfer, and that could impact their properties. So in Israel, we know that when we have dust events, the species richness of microbes increase triple in the number, and we see cell concentration increase 10 times, one-fold higher with this dust. So really an import of new microbes into the environment, a significant carrier. And a lot of studies globally have recently started to check the microbe signature, so the genomic composition of this dust. Basically very easy. You take the dust, you extract DNA, you sequence this DNA genomically, and then you have the community composition. Each line here basically is the relative distribution of different species in this sample, okay? So these are dust events, and these are regular days. Each sample is presenting a regular day, and what is different really in this, but also in other studies of dust microbiome, is that you can see here these green ones and red ones significantly higher in the dust event. So these are species of spore-forming bacteria. Firmicutes in green and actinobacteria, very interesting, very surviving bacteria. The firmicutes, by the way, are very abundant in our abdomen, in our body, but these are different species. The actinobacteria, some of them can contribute to antibiotic formation, so antimicrobial compounds. So they have really interesting secretion metabolites. The problem with these studies that others and also us do, the genomic studies, is that basically we cannot understand what is viable and what is just leftovers of a bacteria. Like the remaining of a bacterium that is already dead, but still DNA is there because DNA is very stable. And this signature might not be relevant to what is really active in this dust. So this is not enough for us to really understand what's happened there.
What's the blue that's decreasing during dust storms?
This blue?
Yeah.
It's not decreasing so much, not significantly. It's the beta proteobacteria. It's part of the proteobacteria phylum, and we do find it in the dust. We do find it. And
I can see it.
but it's a common communities that is always there also in other days. Might be, okay, so this is phylum level, might be that lower taxonomic identity would be different. I show here only the phylum level, which is a bit broader for the story. So in Israel, as you know, we have a lot of dust. We have dust coming from different directions. We have dust coming from north, from south, from Sahara that can go either through the Mediterranean or through the Red Sea, so different paths, and also from the east, all the way from Saudi Arabia. And this dust you can also see in this figure, very different in their colors. Right? You see here, this is a bit more yellowish, this is a bit more brownish, and the composition, the nutrient composition, the chemical composition of the dust is different. So we expect also the microbial community to be different on these different environments. By the way, this one is a bit more anthropogenic impacted because it's transfer, crossing a lot of inland path, and it's impacted. We really see the dust coated with brownish coat. And this is a nice dust day in the Technion rooftop. You see this is dust coming from east. And we sampled this dust with an instrument that we got from here, from Igal. And we have on top of it another instrument. So this is a filter sampler, and we have also the Coriolis sampler, which samples into an empty tube. Basically, we have the dust in a tube, which is really easy then to do many different manipulations later. So we do a lot of different analysis on this dust. We can do chemical analysis, genomic analysis, microbial analysis. All this stuff could give us a bit more broader understanding of these microbes. And what we did initially was just to take a look, just to look at the microbes. If we see them, we label them with a dye that labels dead cells. So the red cells would be dead, but if we find cells that are not labeled, they might be viable. It's not enough proof, but it's still something to hold on, to initially start with. You see these clay minerals in the dust. You see some remains of organics, and you see shapes of bacteria-like forms. So this is really a good indication to try and understand. We have dust cells, bacterial cells in this dust. And then now we really want to know what is viable, what is active. Would they be just spores there, just dormant species, or really active? So this is something we try to understand. So first of all, like everyone else, we did genomic study to extract all the DNA and see we have different community composition for different dust origins. So dust coming from east, dust coming from the Sahara, and another one coming from east with a contribution from the sea. And we see different clusters of the genomic community composition. This was led by Peng Fei, which I don't have the figure, and Johnny. And they did this genomic study. What we did here additionally is to look also, so we have the DNA, and we also looked at the RNA level. RNA is much less stable than DNA. And if the cell is dead, we expect the RNA to degrade much faster. Okay? So it's kind of an indication, initial indication from what was recently living. And from there, you can see here on the top is the DNA community composition, and on the bottom is the RNA, okay? For different phyla, for different species, as we saw in the beginning. You see here that this, for example, proteobacteria have a lot of DNA and also a lot of RNA. And then Bacteroidetes, actinobacteria has predominantly higher active fractions and also firmicutes, like we expected. And this is cyanobacteria, which is also an interesting species. We didn't get into it in this study, but it's in the plant. Then we looked at these active species and their relative abundance of the different species. You can see both day samples and night samples for this species. We were looking for bacilluses, which are from the firmicutes, and other species. And then we tried to see which one is connected to the other, which one is correlated with the appearance of the others. So it's like a matrix of network analysis to see which one comes with the other. And we really see there are some species that tend to come together in the same dust. Maybe there is a kind of a community interactions. This is like a first indication.
What do you make of the differences between day and night? Or are they identical?
I am cautious about it because it's only 3 events. I think we need to do more studies on day and night. It seems like different. I think it relates to relative humidity increase in nighttime, but I cannot say something really strong about it. As you saw in the beginning, as I showed you, so firmicutes, for example, are much higher in the dust event, and we also find them here, much more active also, and specifically Bacillus is a very interesting species in other environments, in plant, in soil, in our gut, and also apparently it's very abundant in the atmosphere. So for that, we took those samples of the dust and heated them to 80 degrees to kill everything that is not spore forming, or not bacillus, not actinobacteria, everything is expected to die. And then we isolated those species using, we plated them on other plates and identified them genomically. So you see here, each one is a different isolate from the dust. And each one was then grown and DNA extracted and identified genomically. Okay? And then we have a collection of dust bacilluses, different bacilluses in the dust. You can see different shapes, different sizes, different colors. That means they are really rich, and this is just one family, just one. Apparently the dust is very rich. We just narrow it down to look only on specific bacilluses, which could be interesting. Now we plan to do it on other species as well, but this is just an initial study. And really genomically, what we did to identify them, we took 2 different regions in their genome and identified them. So in some cases, we had the similar identity. Both regions told that this is Bacillus subtilis, for example. This one. Okay? And others show different identities. So in this region, it was labeled as Bacillus X, and on this label, it was Bacillus Y. And that means it might be a different Bacillus than what is known already. So we really have new species here that we need to identify and sequence the entire genome.
So how do you define Bacillus?
In genomic analysis?
Yeah, if you say there is Bacillus X, Bacillus Y, what's the code?
It's a good question. We sequence basically as a region in the DNA. One is the 16S for general bacteria, and one is very specific for bacilluses. And we have libraries in the internet that are deposited there. People from all around the world deposit their sequences of their cultures or their identified species, and we compare to what is known. We can only compare to what is known. If we don't know, if we have differences, we need to sequence that. It's called de novo sequencing. We need to sequence the genome completely and deposit it, and we can identify its bacillus because of the morphology and spores. But it's a good question. This is an indication only on what is really similar to what is known already. And you can see in blue is samples taken in the fall time. In red are samples taken in the springtime. So we had 2 different seasons. In both of them, very rich community composition of bacilluses. Then we tried to characterize growth rate of these different isolates. And you can see that the different rows are different media type. This is the limited media, and they're really struggling growing there. This is the media rich with calcium, that we suspect calcium would be a good indicator for dust. Also not as good. And this is a rich medium with different nutrients, calcium, nitrogen, carbonate. And you can see also here the growth is different for the species. So each one has its own growth. Some grow really fast, some go really slow. So this community is complex. It's not unified, it's not homogeneous, not species-wise, but also not behavior-wise. And each one apparently has a role there. It's like a niche, a growth niche. That each one has its own strategy for survival, for harvesting food. And then it became interesting. When we saw this, I showed you before the morphology, we saw different colors, right? And the colors indicate metabolites formation. And these metabolites sometimes are for communication, for chemical communication between the species. So what we did here is to test them and to see which is strong and which is weak. And we grew them one next to the other, compared to for the single one. And what sometimes we saw, I show here only the interesting things, that really the presence of the other one lead the first one to engulf it and eat it and really go wild and grow all around the plate. And sometimes you can see degradation of this one, for example. It's really decay also here. That means that there is a chemical communication between them. There is something that stimulates interaction between these species. And if you try to imagine that on the dust, which is limited with organics, that might be the source for their food. So they might succeed if they can have other to predate. It's like a jungle, basically. The strong survives. And here we try to do it a bit more structured. We grew the same species, one next to its own species, so you can see really similar morphology. When it's grown with another one, it stimulates this one growth. And with the third species, it's really engulf it and kill the other one. We tried to do it kind of initial screen for different species. We did like this matrix of selected species against all others, and we ranked it. If it's stimulate growth, it's called it agonist, so it's labeled in blue, and if it's inhibit growth, it calls inhibitor, like predator, and if it's not impacting anything, it's neutral. And you can see here different behaviors of these species. Some of them are really strong, like this one, for example. It really kills everything. And that is really nice, but it's really on the plate. It's really a very initial statement to say they have this ability, but would it be also in the dust? We cannot still say that from here. What we did then was to take them and really prove that it's because of chemical communication. We grew each one with a conditioned medium of the other. So if you grow them both in liquid media, and then you take out all the bacteria but leave only the liquid solute that could contain also the metabolites from this bacteria. And then you could expose the other only to this liquid. So what you can see here is this species, the bee, without anything alone. And then the bee itself with its own conditioned medium, with no significant difference between them. But when it's exposed to the conditioned medium of the second one, it really inhibits the growth, and it's really significant. On the other way around, this species, you can really see that it's a bit stimulating the growth with the presence of the second culture. So this is another indication for this interesting phenomena. It could be antimicrobial or even stimulator of growth, which could be interesting for many applications. But in our case, in our story, that could mean that this is serving them as a tool to survive on the dust. What we then did with the Ella here that led this study, she is interested in carbon fixation. And what we see in these isolates, you can see here, this is the biofilm basically of this dust, of this bacteria, bacillus. And it has folds. You see these folds in this biofilm. And from other studies from plants, we know that some bacilluses can do biomineralization of calcium carbonate. And we try to see if we also have that in our isolates, because we speculate that that could also serve as a protecting mechanism for them to form this crystal of calcium carbonate to protect from UV, for example. And what we did was micro-CT. Micro-CT is basically like a CT. It scans density differences between soft tissue and dense tissue. And you can see here in this composition figure that these yellow ones are the one with the dense structure, which speculates to be calcium carbonate, as we know from other studies. It's not enough yet. We now do FTIR to try to understand which type of crystal is it. Basically, it's your field, right? It's more from your studies. So it's really a good topic to collaborate on. If someone is interested, I'll be happy to hear your input. And then we really took it to the next level and tried to see what is happening while these bacteria are on the dust. Okay, so in the lab, it's not enough. We see them in the nice warm cultures with humidity and no UV, it's not enough. What is really happening on the dust? Can they go there? This is the proof of concept really still needs to show. So we started to see the dust we have and try to understand the composition of elements in the dust using SEM-EDS. And we really see a peak of the calcium, silica, alumina, as we expected in dust. It's comparable to other dust composition. Then we took a species of bacillus that we grow in the lab. It's a lab strain. It's not the one that we isolated from the dust. We have labels. We have tags on this species genome. So for example, if it will sporulate, it will secrete a protein called adhesin that attach to its surface and form a spore. And then it will be tagged with red labeling. So red labeled bacteria have adhesin protein expression. Okay? The green ones are expressed for the flagellin, which is a protein for the flagella, for the tail of this bacterium. That means this bacterium is motile, is not dormant, it's really active. So green ones are the ones that we're interested in, that are active on the dust. So you can see here the figure of the 24 hours incubation on the dust. And this is 48 hours. You can see really the spores formed, but really still we have the green labeled, and I'm sorry if someone is colorblind. This is not my selection of colors, but it's really abundant. This really green active bacteria are here also after 72 hours, a bit less, but still active. So theoretically, a dust carrying bacterium that survives there could be staying alive for, according to this, for 74 hours, which is not a small amount, a small time, short time. And then we also challenged this bacterium and tested nutrient preference. So we have the dust bacterium, the dust enriched with glutamine, which is a nitrogen source, and the dust enriched with glycerol, which is more preferred. From this figure only, you can see the glutamine, right? The nitrogen source is apparently lacking in the dust. So carbon, they can handle that. But nitrogen, if you increase nitrogen content in the dust, it's really a good thing for them. So that's also strengthened the idea of them knowing how to predate and how to get their organics from the dust, as they don't seem to mind the glycerol addition. Then we further took our isolates, not labeled for any tag or something, but here it's really confusing, but the red-green labeling here indicates live dead. Okay, live are green, dead are red. And this is also not my selection of colors, it's just a kit that we buy, and we grow these isolates. We took on a dust that we sterilized. We took the dust, we sterilized it, and incubated it with the bacterium we isolated from the dust. And then you can see really happy growing dust bacterium, but then we add the glycerin as well also here. You really see happier culture on the dust. And then what we did was to take this dust and to remake a new synthetic dust in the lab. We screen the composition, the chemical composition of the dust, and we make a dust in the lab without any organics, okay? With calcium carbonate, with clay minerals, with iron, silicates, and everything, and sterilized it, and grew the same culture on natural dust, which is sterilized, compared to the synthetic dust without any organics. And you can really see the difference in this graph, and I hope you're convinced, really, the organics there is really a substantially important compound for the bacteria to survive. And here you can see it in a confocal microscope, really seeing the labeled bacteria on the dust particles. This is natural dust. This is synthetic dust without any growth, not significant labeling at all. This is very exciting for me because here what we did is to take the sterilized dust as is without any addition of a culture and just take it into a humidity chamber. Just make it happy, make it with humidity. So it's basically have nutrient, organics, humidity, and it's grown. And you see really bacteria growing on the dust compared to the synthetic dust. And this is in SAM, you really see bacteria agglomerate. Maybe some of you can relate to other bulbusim, how do you call those structure in the nature? So it's really similar to that. This is the dust, and this is bacteria growing on it. So really, this is ongoing study. We have every day new results to understand, to understand this story better. But basically, this is a very interesting topic. We find a lot. What we did next was to take the dust and use labeling for calcium in entering to the cell. So calcium biomineralization, we want to see that. We took the bacteria and grew them on the dust. So this is the dust particles. And this is the dust with the labels of the calcium MA, which is a dye that labels calcium in the cells. It has to pass through a cell, through the membrane, to be labeled. So if it's out from, it's not labeling the dust calcium, which is really important. It's only labeling the bacterium calcium. And this is the overlay of the bacteria on the dust doing some nice things with calcium. So this is really an ongoing study, as I said, with Ella that tried to resolve that. And maybe to summarize that into a figure, we have the dust, as we understand that, with the bacterium and all the microbes on it. And it has its nutrient, it has interaction with other species on the dust. Experiencing stress in the atmosphere, different humidity, different gradients of temperature that get them, those that will survive these stress conditions are very interesting for us to understand them. What enables them to survive that? What are the properties that they have to survive? And they show you today that these communities have difference in the growth rates, different behavior, different excretion of metabolites that could help them predate or protect themselves against others. They might also have calcium carbonate crystals in them that might help them against UV radiation, which is still under study. But the most important thing for me here is that we managed to show a proof of concept of these dust microbes growing, really actively growing on the dust at the growth medium. So the next thing would be to understand their impact, how significant those microbes in the atmosphere, how much we have them, what are their activity there, if they do calcium carbonate biomineralization, what is the rate of calcium, of CO2 interact with the calcium there. That is really interesting for us to understand. So I would like to thank you for listening. I would like to thank Ilana Kolotkin-Gal from Reichman, which is an expert on bacilluses, and we collaborate on this topic. This is my group with the new incomers and in Haifa in the Carmel Mountain. You're welcome to come there and visit. And thank you for your attention.
Thank you.
I would like, just a minute, just to highlight a conference. Some of you get my email, but if you don't get my emails and you want to come to the aerosol conference in March, please contact me. We have a registration. It's really easy to come with public transportation, train, and then a cable card all the way to the Technion. That's it. Now questions.
Questions?
You showed us that trajectory that assume. Some of them go, I assume, very high.
Right.
So the dust particles in those back trajectories might have experienced very, very low temperature.
That's right.
So what happens to the or?
But we
those
don't know.
atoms?
We don't know. We have now a chamber that we want to simulate that. And the plan is to really take microbes that we have here and lift them up to really high temperature, high levels, high elevation, to see what is the effect. But we still cannot say. It's really an interesting question we ask ourselves. We think that those bacteria, they can produce biofilm, right, and spores. So this biofilm is a protecting medium that can help them to protect against freezing. They also may be the metabolites themselves, anti-freezing metabolites. We still don't know, just speculations.
So will they survive sub-zero, sub-freezing temperatures?
Apparently, when they come to our lab, our vial, we can grow them. So something grew there and remain alive.
Because they probably experience temperatures way below the freezing point. No, the dust particles.
I mean,
traveled thousands of kilometers. They must have gone a few
some of the
kilometers
dust
up.
events really lift all the
A few kilometers.
Yeah.
So that usually accounts for drop of below the freezing point.
Yeah.
So okay, you don't know the temperature.
No, I don't know, but I know that we in the lab we freeze those bacteria and then we can recover them. They can grow. So it's not yet any.
So I'm thinking of Saharan dust plumes. So they can travel days, weeks, all the way across the Atlantic. Do you know, speculatively, do you know whether these organisms can change the albedo of these dust plumes?
If they interact with like water droplets, I guess that would be significant. And I know some of them can be ice nuclei bacteria. Some of them could be cloud condensation nuclei, fungal spores, for example. But I don't know if they themselves could change the albedo. I don't know.
It could be an interesting feedback mechanism of organics and.
So there are some studies on like brown carbon and like organic carbon, their interaction and their impact on the albedo. It's a good question. I don't know. It depends, I guess, because if there are spores, they also have a different color, so more dark. If they are not spores, they will be whitish and yellow. So.
Especially in the Atlantic, because these dust storms affect all the way from the occurrence of hurricanes to convections, even the position of the ITCZ, lots of interesting feedback. So if they have an effect on these dust storms, it could be a nice way to link that to climate change or just climate variations.
It's a good question.
So what's the potential of these bacteria as they travel out of plants to actually change and influence the bacterial communities that are being introduced into the soil, etc.?
So there are a lot of studies on that. So in ocean, for example, we don't look at them. We look at the dust itself, how that fertilizes the ocean. Also in soil land. There are, I think, studies that showed transfer of pathogens and import into a new environment. I don't know if there are some studies that are broad enough to really take into account the active fraction, how that impacts. So we know that pathogens, plant pathogens, are transported from very long distances. So this could be one of the sources. We don't have any proof for that yet.
Is it only filtration of the initial community, the ones that are surviving, or there are new creatures that are growing? And when you see, I understood that you saw it only in the experiments, but when you see the stimulus growth of the other kinds, like, do they eat the other? Like, what stimulates the growth?
We think that it's chemical communication, that we saw when we filter out the bacteria, we still see the increase in the growth of the others. So it's kind of chemical compounds, metabolites that are secreted out and induce some difference in behavior. The first question was about others that join the dust along the way. Mm-hmm. This is something I ponder a lot on. So I guess dust is a dynamic environment. It experiences differences, and then along the way it can take with it some bacteria or fungi. That is, the thing is that the concentration of the microbial community without any particles on them in the atmosphere is much lower than this is that attached to a particle. So the one that's coming with the dust itself in the origin would be the major community, and then some could come along the way, but the concentration of these cells, we count them in the atmosphere, it's much more reduced, like order of magnitude reduced compared to the one we see in the dust. So might be, but not significantly.
So the diversity, just the diversity is higher in the atmosphere and the dust?
The dust increased diversity significantly. Okay.
How do you care for that your station is on the roof of the Technion? Because I mean, do you try to compare it with other stations? For example, this instrument that they're using, Eagle was using here on the roof of our building. And the question is if you have any kind of intention to do this comparison, because Haifa is overall an area where there are a lot of pollution.
We go also to Eilat. We now sample in Eilat and in Haifa.
Okay.
And in Eilat, I hope it will be much clearer and cleaner because the dust is directly coming from Sahara. In Haifa, what is good is sometimes we have the dust coming, it's more complex. If it comes from the Mediterranean Sea directly, so it's more, the signature of the dust is more clear. If it comes from inland, I don't like these samples. It's really impacted by a lot of organics, local organics, and others.
So you sample only the dust storms that go over the Mediterranean?
We sample all of them, we compare them, but we rely dust communities on those that are clean from terrestrial.
More questions? If not, then thank you again.